Welcome to Animal Biotechnology. Today, we are going to explore a fascinating and powerful molecular technique known as RFLP-based diagnosis. Imagine being able to look deep inside a person's genetic code and spot a single tiny error long before any physical symptoms of a disease ever appear. This is not science fiction. It is a foundational tool in modern biology. By the end of this lesson, you will understand exactly how scientists read the hidden barcodes of our DNA to diagnose severe genetic diseases. Let us dive in. To understand how this works, let us start with a simple analogy. Imagine you have a long piece of ribbon, and you are told to cut it with scissors every time you see the word "stop" printed on it. If your ribbon has the word "stop" printed 3 times, you will make 3 cuts and end up with 4 smaller pieces of ribbon. Now, imagine your friend has a similar ribbon, but a manufacturing error changed one "stop" to "slop." When they cut their ribbon, they will miss that cut and end up with fewer, longer pieces. This simple idea of cutting at specific words and comparing the lengths of the resulting pieces is exactly how scientists diagnose genetic diseases. In biology, this ribbon is our DNA, and the scissors are special proteins. Now that we have our analogy, let us look at the formal name of this technique. It is called restriction fragment length polymorphism, or RFLP for short. I know that sounds intimidating, so let us break it down into bite-sized pieces. First, restriction refers to restriction enzymes. These are the molecular scissors we just talked about, which cut DNA only at highly specific sequences. Next, fragment refers to the pieces of DNA that are left over after these restriction enzymes have made their cuts. Just like the pieces of ribbon, these fragments are the physical material we will be analyzing. Continuing with our breakdown, the next word is length. This simply means the size of these DNA pieces, which scientists usually measure in base pairs. Finally, we have polymorphism. This comes from Greek words meaning many forms. In genetics, it refers to the natural variations or mutations in DNA sequences among different individuals. Putting it all together, RFLP is a technique that detects genetic differences by cutting DNA and comparing the sizes of the resulting fragments. If there is a mutation, the cutting pattern changes and the lengths of the fragments change with it. You might be wondering, why does this concept matter so much in the real world? RFLP was actually one of the very first tools scientists used to literally see variations in human DNA at a molecular level. It is absolutely crucial for diagnosing severe genetic diseases, such as sickle cell anemia and cystic fibrosis, long before a patient shows physical symptoms. By identifying the exact genetic mutation, doctors can provide early treatments, and parents can understand the genetic risks for their future children. Furthermore, RFLP laid the foundation for DNA fingerprinting, which revolutionized forensic science and paternity testing. Without this brilliant technique, we would be completely blind to these hidden genetic mutations. Now that we understand what RFLP is, let us look at the mathematics behind it. When we cut a linear piece of DNA, we can predict exactly how many pieces we will get using a simple formula. The formula is n equals s plus 1. Conceptually, this just means that the number of fragments will always be 1 more than the number of cuts you make. Here, n represents the total number of DNA fragments produced. S represents the number of restriction sites, or cutting locations, on a linear DNA molecule. If a mutation increases the number of restriction sites, S, the total number of fragments, N, will also increase, and their average length will decrease. Let us apply this formula to a concrete example to build your confidence. Suppose we have a linear DNA sequence that is exactly 1,000 base pairs long. We are given that in a healthy individual, a specific restriction enzyme recognizes exactly 3 cutting sites on this gene. We want to find out how many fragments are produced. We choose our fragment calculation formula because we are dealing with linear DNA. We substitute S equals 3 into our equation. Adding 1 to the number of cuts gives us the total pieces. The final answer is 4 distinct DNA fragments. Physically, this means if we run this healthy DNA in a lab, we will see exactly 4 separate pieces. Now, let us see what happens when a disease is present. In our patient, a genetic mutation has destroyed one of the original cutting sites. We need to calculate the new number of fragments. First, we determine the new number of restriction sites. Since 1 was lost, S now equals 2. We use the same formula, N equals S plus 1, because the DNA is still linear. Substituting 2 for S, we find that the restriction enzyme is only able to make 2 cuts. This results in a final answer of 3 fragments, which is 1 less than the healthy individual. By simply counting these fragments in a lab, a scientist can immediately tell who has the mutation. This brings us to another fundamental rule of RFLP, which is the conservation of DNA length. Conceptually, this means that cutting the DNA does not destroy any of the genetic material, it just divides it. The formula is L total equals the sum from I equals 1 to N of Li. Let us define these symbols. L total is the total length of the original uncut DNA molecule, measured in base pairs. N is the total number of fragments. Li represents the length of each individual fragment. The summation symbol simply means we are adding up the lengths of all the individual pieces. If a mutation destroys a cutting site, 2 smaller fragments merge into 1 larger fragment, but the total sum remains exactly the same. Let us apply this conservation principle to a real-world medical diagnosis, sickle cell anemia. This disease is caused by a single letter change in the beta-globin gene. We are given that a restriction enzyme, called DDE1, normally cuts this gene into several pieces. In a healthy person, the enzyme successfully makes a cut, producing 2 specific adjacent pieces. The lengths of these pieces are 175 base pairs and 201 base pairs. We are establishing this healthy baseline first, so we know what normal DNA looks like. Physically, this means a healthy person's DNA will show 2 distinct smaller bands when tested in the laboratory. Now, let us calculate what happens in a patient with sickle cell anemia. We're given that the mutation destroys the specific DDE1 cutting site that separates those 2 pieces. We need to find the size of the new fragment produced. We choose the conservation of length principle because the total DNA material is unchanged. Since the cutting site is gone, the enzyme skips that cut, and the 2 fragments remain joined together as 1 single larger piece. We simply add their lengths together, 175 plus 201. The final answer is exactly 376 base pairs. This shows exactly how a tiny molecular change translates into a measurable mathematical difference. Let us look at how these mathematical results actually appear in a laboratory. What you are seeing here is a diagram of an RFLP gel electrophoresis. The vertical axis represents the DNA fragment size in base pairs, with larger fragments sitting higher up. We have 3 columns. On the far left, the normal individual shows two distinct bands at the bottom, representing the 175 and 201 base pair fragments. On the far right, the disease individual shows a single heavy band higher up at 376 base pairs because their cutting site was destroyed. In the middle, a carrier has both the normal and mutated genes, so they show all three bands. This visual perfectly illustrates the power of RFLP in clinical diagnostics. We have covered a lot of ground today, so let us review the key takeaways. First, restriction fragment length polymorphism is a powerful tool that turns invisible genetic mutations into visible, measurable differences in DNA fragment sizes. Second, we learned that by using restriction enzymes as molecular scissors, we can cut DNA and use simple addition to predict the sizes of the resulting pieces. Finally, we saw that when a mutation alters a cutting site, the fragment lengths change, allowing doctors to diagnose severe genetic diseases like sickle cell anemia with absolute certainty. You now have a complete foundational understanding of how molecular biologists read the hidden barcodes of our DNA. Great job!